Today I'm going to give a talk about the social microbiome gradient across the rural to urban transition in Indonesia. So what is the meaning of social microbiome? It's the collective microbial meta-community of a social group or network. We use this term because we studied the nature of the microbiome at the social or social group or community scale. So here we can see that Indonesia is very large. It's a large country. It's a maritime country, and the population is the fourth largest in the world, over 283 million people, and among the most diverse regions worldwide, with over 1,000 of ethnic groups officially recognized across the archipelago, speaking more than 700 languages. However, despite this, Indonesia is presented only a very small percentage, 0.01% of global metagenomic samples. So our previous study in the Balinese from Denpasar revealed that there is a distinct community structure of the Prevotella rich versus Bacteroides rich. Balinese from Denpasar, on the right panel, Balinese from Denpasar colored as red, clustered between the pre-industrialist Hadza hunter-gatherers, Malawians, Guahibo, and Amerindians in the green color, and industrial country like Americans and Italians colored in blue. Balinese urban population, this is the samples that we collected from Denpasar, a metropolitan city, the capital of the Bali province. So, however, the Balinese urban population has relatively high Prevotella versus Western urban and appears to be transitional among this pre-industrialist and industrialist society. Okay, and then after the Bali study, we got, of course, that's one of the downside is you have to have a grant, international grant, to be able to fund it yourself. Otherwise, it will be very, very, very expensive to study the microbiome. We are lucky to have support from several grants, and we started the study of the Indonesia Microbiome Ecology and Evolution. This is mainly supported in collaboration with Cambridge University, the project what we called Mobile, we got the grant from European Commission. The Indonesia Microbiome Ecology and Evolution is abbreviated as Indomie. Indomie is the famous instant noodles food in Indonesia. That's why we call it a nickname, Indomie study. In this study, we included communities that occupy the islands, 3 islands, Bali, Borneo, Kalimantan, and New Guinea or Papua here, spanning an archipelago of approximately 2,500 kilometers. All tropicals, living in tropical climate and interconnected recent and more ancient human histories. Critically, the group also spanned a range of lifestyles, some of them living in the transitional stage like Bali, some of them are still living in the middle of the forest, and some are fisher horticulturalists such as the Asmat Daikot in the Papua, and also mixed forager agriculturalists, Punan Aput and Punan Tubu in Borneo, in Kalimantan, horticulturalists also in Kalimantan, the Basak people, agriculturalists Balinese in Pedawa, the rural Balinese, peri-urban residents, Punan Tubu Respen, who relocated in the 1970s near this small town of Malinau from the forest, and then fully urban residents, Balinese in Denpasar. Collecting fecal samples in remote and diverse locations require detailed logistical planning to overcome challenges like limited access, lack of electricity, and non-standard transportation. So we heard that Adrian said that as soon as possible, you should freeze your samples. It is not possible for us because some of the area does not have electricity at all, and we have to travel. For example, on the left panel, we travel by a small airplane. In the middle, we travel by boat, speedboat, and also the condition in the field is also varied from one village to another. But we are very happy. We are lucky to be able to do this sampling For your information, we preserve our samples whether we like it or not. We use the very expensive one, preservation, because we want to be able to save our samples until it reach our lab. But that's the reward of that effort is that how much novelty is there in our Indomie dataset? It's very, very, very diverse. And we are able to see when we compare that to the similar, we use the SMP database of Singapore, SPMP, because it's located near our study area near Indonesia, and it has three ethnic groups in their study: the Indian, the Chinese, and the Malay. And from this, we could see that similar size, and also the size is similar. A diverse set of bacterial species in Indomie was identified. It's more diverse than the Singaporeans. There were 191 novel species level and subspecies level. 50% of those 191 novel species are subspecies. Novel max metagenome assembled genomes are widely distributed across the bacterial phylogeny. So how novel is Indomie actually? Indomie effectively captures regional diversity and harbors many village-specific novel microbial species. Novel species are rare and tend to be low prevalence, often found in a single village. So it's not spread. Novel species are not similar from one village to another. Sometimes it's specifically found only in a single village. A bacterial spore formation is characteristic trait of Bacillota. One of the traits that we noticed that in this diverse database of ours, the non-sporing bacteria, so it's not Bacillota, is more spread around the remote and rural area. So we divided this in three. I will show it to you later. And among the sporulation, Bacillota is the one that have a sporulation trait to spread. So among Bacillota, the number of genes involved in sporulation correlates with prevalence, likely highlighting the role of sporulation in transmission. So to see how our data is situated, when we compare that with the Singapore database, the SPMP dataset, we plot that with our gradient of remote, rural, and urban. We can see, as you can see here, that the Indonesian microbiota had higher diversity and richness compared to the Singaporeans. A gradual significant a gradual and significant decline of species composition was observed from remote to rural to urban groups. Individual cluster community also showed that. So this is rural, remote, rural, and urban in the first PCA. And then there is also a broad family level shifts. If you can see this at the bottom of the bottom panel, broad family shifts associated with urbanization. We found in our study that community lifestyle, more than individual lifestyle, predicts microbiome differentiation in Indonesia. It's not individually different, it's a community level differences. We investigated further the drivers of this lifestyle signal by analyzing individual level, diet, and daily activity data that is available. So this is only 7 core food categories: rice, sago, pork, egg, milk, fish, and chicken, and 2 daily activities: hunting, because some of our population are still doing hunting and gathering, hunting and watching television. The PCA of the expanded lifestyle reveals substantial variations. And also there's a strong correlation between microbiome and lifestyle distance between sample pairs, reveals a highly significant correlation between population, but not within population. On the right-hand panel, you can see that within population is no difference, but between populations, the difference is very distinct. On the left-hand side is the graph showing the sporulation and non-sporulation composition of the MAC, of the metagenomic assembled genomes. And non-sporulating are more likely to be observed within a single village, as I mentioned earlier, than across multiple villages. So non-sporulating MACs, the red histogram is the non-sporulating one, are more often unique to a single village than sporulating MACs, the yellow outline histogram, which are more ubiquitously shared across villages. So the sporulating one could be found in various villages. Sporulating or non-sporulating is discriminated by the presence or absence of the master on the right-hand panel of the master regulator gene essential for sporulation, the SPO0A. So what is the take-home message of this presentation? It's in line actually with the aim of the microbiota vault and MVIF, that to preserve this diverse microbiota from the pre-industrialized populations. So what we found that our diverse gut microbiome sampling from Indonesia substantially extend known microbial genome diversity. Oh, sorry, my camera is disconnected. Can you see me now? Okay, I will just continue, and this is my last few slides. We identify a rural to urban climate across multiple levels of biological organization between community, but not within community. Variation in diet is strongly predictive of microbiome composition, so between community, not within. Offering compelling evidence that microbiome divergence is driven by community-level differences, not by individual-level differences. Novel MACs are significantly depleted for sporulation genes. Diets pointing to the role of microbial traits, specifically sporulations, in revealing host microbes' dependence and in shaping microbiome structure. Our work highlights the interplay of host lifestyle, population structure, and bacterial physiology in shaping microbiome diversity and biogeography at the key scale of human communities. So this is really one of the examples that showed how lifestyle climate could affect the microbiota composition. This study emphasized the importance to preserve microbiota from traditional populations. I think I would like to acknowledge the whole team. This is our team before and after the Eijkman Institute. Now we are at the Mohtar Institute for Nanotechnology, and these are our collaborators. Without them, this study will not be able to be conducted, and we will not be able to have these interesting and compelling results. So thank you, everybody.